Have you ever wondered how scientists actually categorize, well, every single living thing on Earth? It's a monumental task. Biological classification is basically the ultimate filing system of nature. And getting a grip on it? That's your key to decoding life itself. Let's dive in. Just imagine trying to sort millions of unique life forms. We're talking everything from microscopic bacteria to massive blue whales, all into a neat, logical system. No absolute taxonomic chaos allowed. So how on earth is this immense diversity organized into just 5 categories? Well, that's exactly what we're going to figure out today. Here's our roadmap for this explainer. We'll start with the quest to classify, then look at the 5-kingdom system. After that, we'll break down Kingdom Monera, Kingdom Protista, Kingdom Fungi, and finally, Plantae and beyond. You know, since the dawn of civilization, humans have instinctively classified organisms. Aristotle was actually the first to use a truly scientific basis for this. He just looked at simple morphology, sorting plants into trees, shrubs, and herbs, and animals into those with red blood and those without. Later on, Linnaeus formalized a 2-kingdom system, Plantae and Animalia. But it wasn't until 1969 that R.H. Whitaker proposed a massive, game-changing breakthrough, the 5-kingdom classification. Now, you might be wondering, why did Linnaeus's early 2-kingdom system fail? Well, it completely lacked cellular nuance. It lumped fundamentally different organisms, like prokaryotic bacteria, right in the same bucket with eukaryotes. It also grouped photosynthetic green plants alongside completely non-photosynthetic heterotrophic fungi. Why? Simply because they both possessed a cell wall. Honestly, it was just an inadequate system for understanding the real complexity of life. So, the elegant solution to this massive biological headache? It came in the form of a single number: 5. R.H. Whitaker's system completely revolutionized our understanding of evolutionary relationships. Whitaker really established a brand new scientific standard here. He didn't just look at one random trait. Instead, he evaluated cell structure, body organization, the presence of nuclear membranes, and, absolutely crucially, their mode of nutrition. By clearly distinguishing between autotrophs and heterotrophs, as well as prokaryotes and eukaryotes, he defined 5 distinct kingdoms: Monera, Protista, Fungi, Plantae, and Animalia. Moving on to our first major group, Kingdom Monera, the prokaryotic pioneers. What's really fascinating here is that all bacteria, which, by the way, are the sole members of Kingdom Monera, visually boil down to just 4 fundamental shapes, despite their highly complex metabolic behaviors. We've got the spherical cocci, the rod-shaped bacilli, the spiral-shaped spirilla, and the comma-shaped vibrio. It's this beautiful visual simplicity hiding some incredibly complex biological machinery. Within Monera itself, there is just a staggering level of metabolic diversity. Take Archaeobacteria. These are the extremophiles. They literally thrive in the harshest habitats imaginable, from extreme halophiles in super salty areas to thermoacidophiles surviving in boiling hot springs. Then we have the eubacteria, or true bacteria, characterized by a rigid cell wall. Now, while some of these are autotrophs, the vast majority are heterotrophs, acting as absolutely crucial decomposers or sometimes severe pathogens. For a great example, consider the cyanobacterium nostoc. Often called blue-green algae, these little guys are photosynthetic autotrophs, and remarkably, they can actually fix atmospheric nitrogen using these specialized, slightly larger cells called heterocysts. They play a massive role in recycling environmental nutrients. Talk about punching above your weight class. But hey, there's always an exception in biology, right? Enter mycoplasma. These are literally the absolute smallest known living cells. Unlike true bacteria, mycoplasma completely lacks a cell wall, and get this, it can survive entirely without oxygen. Many of them are highly pathogenic, proving that you definitely don't need a rigid structure to be a formidable organism. Next up, Kingdom Protista, the single-celled eukaryotes. The boundaries of Protista are, well, notoriously blurry. What one biologist calls a plant, another might actually call a protist. This kingdom acts as a vital evolutionary link, spanning 5 distinct and incredible groups: the cruciferophytes, the armored dinoflagellates, the super-flexible euglenoids, the saprophytic slime molds, and the predatory protozoans. Visually contrasting 2 of these protists reveals some serious evolutionary adaptation. Take euglena. It's a flexible mixotroph with a protein-rich pellicle.
It happily photosynthesizes in sunlight, but throw it in the dark, and it hunts like a heterotroph. Meanwhile, a ciliated paramecium uses the coordinated movement of literally thousands of tiny cilia to actively steer water-laden food directly into its gullet. It's like a microscopic vacuum cleaner.
And we definitely can't forget the diatoms, which belong to the chrysophytes. Their cell walls are embedded with indestructible silica, making them look kind of like microscopic soap boxes. Over billions of years, these tiny shells pile up on ocean floors to create what we call diatomaceous earth. Because of its gritty nature, we actually harvest this ancient dirt for things like polishing and filtering oils. How cool is that?
Let's shift gears to kingdom fungi, the great decomposers. You know, for the longest time, fungi were mistakenly just lumped in with plants. However, while plants are autotrophic with cellulose cell walls, fungi are strictly heterotrophic. They absorb soluble organic matter as saprophytes or parasites. And crucially, their cell walls are composed of a super tough material called chitin. So Whitaker's system finally gave them the respect and the unique kingdom they deserved.
Now, fungal sexual reproduction can seem tricky, but it logically breaks down into 3 beautifully synchronized steps. First, we have plasmogamy. That's the fusion of protoplasms between 2 gametes. Second is karyogamy, the actual fusion of the 2 distinct nuclei. And finally, meiosis, the reduction division in the newly formed zygote, which results in haploid spores just waiting to grow. But actually, hold on, there's a fascinating exception that occurs here called the dikaryon phase. In certain fungi, like Ascomycetes and Basidiomycetes, the nuclei don't fuse right away after plasmogamy. This creates a temporary intervening stage, represented as N+N, where a single cell houses 2 completely separate parental nuclei before they eventually fuse. It's like they're just roommates for a while. When fungi reproduce, they form these incredibly complex fruiting bodies. And the sheer structural diversity here is wild, ranging from the molds of mucor and aspergillus to the familiar agaricus mushrooms you might see on a pizza. This diversity, alongside the morphology of their mycelium, forms the exact basis for how scientists divide this kingdom into distinct classes. By looking closely at those structures, we get 4 distinct classes. First, the aquatic phycomycetes. Then the ascomycetes, or sac fungi, which includes super beneficial things like yeast and penicillin. Next are the basidiomycetes, covering mushrooms and puffballs. And finally, the deuteromycetes. These are mysteriously named the imperfect fungi, simply because, well, only their asexual or vegetative phases are currently known to science.
Moving on to kingdom plantae, the autotrophic foundation.
Now, even in a kingdom that's strictly defined by its prominent chloroplasts and its ability to photosynthesize, nature still loves to throw us a curveball. Plantae actually includes partial heterotrophs, like the insectivorous Venus flytrap, and even parasitic plants like Cuscuta. It just goes to show that biological categories are rarely perfectly black and white. So, as we wrap up this taxonomic explainer, I want you to ask yourself something. Whitaker's 5 kingdoms are brilliant, sure, but they completely exclude acellular entities like viruses, viroids, and prions, not to mention complex symbiotic organisms like lichens. They just defy these traditional boundaries entirely. With new genetic discoveries happening quite literally every day, how long will it be before we have to redraw the entire map of life all over again? Keep that curiosity alive, and thanks so much for joining me on this explainer.